coming up with you.
Copy that. Looks like the slope is increasing a little bit right here.
Yeah.
Maybe just local, but...
Still looking back at you.
Mm-hmm. Copy that. Still pushing ahead.
Oh, there's another sea cucumber on the right here.
Oh, it's a different one. I wonder if we could take a quick snap
Quick zoom on
of
that
it.
video. Let's see.
We saw this the other day, didn't we? This sort of...
Yeah, he's got the round...
Oh,
he's going to take off.
It looks kind of pillow-padded-like.
He's got those round ossicles or something in him.
Oh, those are tube feet, I think.
2 feet? The white things?
Yeah, I think so.
Oh, okay.
I mean, they're modified. So you know how some sea cucumbers just have them along the entire length of their body. I think these might be modified on different sides. Or it could be scleroids. You might be right. Huh, interesting. It's got that fringe around the edge. Sort of reared up for a minute, but...
Yeah. Okay, video.
Yeah, I think we're...
Okay, pushing ahead.
Thank you.
So this is the guy. He's in. He's
semi-lacted.
He's not in the animal guide yet, but he'll appear in the next version of him. This is actually seen in the Mariana.
Oh wow.
Mariana area, Marianas Islands.
One of the things about deep sea echinoderms is you often find them present over very large areas. It's unclear yet if it's exactly the same species, but the morphology that distinguishes them from one another is often very indistinguishable.
Yeah, so the identification, this is Dr. David Paulson, who's at the Smithsonian Institute, who works with Chris there, and he's a Holothroidian expert. And he was only able to go to the family Synallactidae, for this particular guy.
We should not have told.
Well, sometimes pictures are difficult means of determining species identifications, but
like all the time, almost.
all the time, yes.
Sparky.
And just to clarify, we were an institution, not an institute.
I keep saying that.
I
Sorry,
know.
institution.
It's simply one of those little things.
Whenever you think you're far enough out ahead, try to
a little
start
bit
covering
more,
some ground.
Get
A
us centered
couple
up
more
here too.
sponges up ahead, and there's a Bathypathes black coral that we've imaged a couple times on this cruise so far. That's the orange one that you'll be seeing at home there in just a few seconds.
I think we'll want to do a little more west, maybe like 220. Maybe.
The more west, the
This is
better
the
my
sort of
waypoint,
leafy one
so,
that's standing
whatever you
upright.
like.
Looks like
Okay.
it looks like more a straight shot up.
It's like a bunch of weird things
Yeah,
going on the tip of it.
Yeah.
Barnacles,
225.
I think.
Yeah. Pilot, would we be able to do a snap zoom on the tip of the sponge?
Sure.
Thank you.
I think you go ahead and get it in, Dave.
All right.
Just do a quick
Okay,
zoom.
video. Zoom in. Let's see if I can.
Bridge nav.
Oh, hydrozoans, I guess.
Interesting. Yeah.
This is the bridge.
See
Go
if
ahead.
I can hold this here.
Another move, please.
So,
30 meters. Bearing
We've
225
discussed
degrees,
in the past how
0
many of these
decimal 2
corals and
knots.
sponges are kind of themselves homes for other animals, especially
30
when
meters,
they die
bearing
and their skeletons are
225,
left
speed
behind,
0
get sort of
decimal
inert.
2.
This is one of the many types of animals that finds itself sort of colonizing other animals. These are
Nice shot.
hydroids or hydrozoans. These are the same type of animal that you find in your sort of dish of water at a freshwater pond, except in the deep sea, there are many, many, many of them. Sometimes they're quite large, and sometimes they're small and form these very abundant colonial forms. But they're very
Don't rotate too
difficult
much
to
further that
identify
way.
and
Okay. You got a little
present
stock coming up.
widely.
Sorry, I didn't want
Okay.
you to hit
Yeah.
You'll see it
No, I
coming.
see it.
Yeah,
Oh, there
okay,
it is.
okay.
Yeah, thank you. Yeah.
ROV nav. Move initiated.
Thank you, Bridge.
Oh, this thing right in front is a dead sponge stock, and there's several dead sponge stalks around here.
Did that get what you needed there, Watch Lead?
Yes, it did. Thank
Okay.
you very much,
Thank you.
Pilot.
Thanks, Chris.
Yeah, sure.
Tina Molotova from the Shirshov Oceanographic Institute identifies those hydroids as something like the genus Solandaria.
So what did we say it was 220?
225.
225. Okay,
Yeah.
copy that.
I'll
That's
head
a
over to
pretty
that
long
right now.
sponge stock there that's fallen down, long dead animal. So sometimes these sponges can get quite high before they finally either topple over or die, or who knows what happens to them.
I'm right on 225 for
Okay.
reference.
Copy that.
Is that just a rock
There's a
formation
weird
to the left?
texture down there,
Yeah,
Pilot,
it looks like a dead sponge.
but it's a huge
It's
dead
big, yeah.
sponge. If you were headed that
I'm
way.
going over that way.
Okay. Wow. It's not going to beat the minivan sponge, though. Ain't going to beat that
Oh, the one that we saw.
in 2015.
few years ago, yeah. Wow, that's quite large, isn't it?
This is maybe more Volkswagen buggish, I suppose, than
Yeah.
Yeah,
maybe not quite that big. Slight exaggeration.
maybe it's.
And
a
it may even
meteorite.
be two sponges here.
Is it alive still?
No, it's dead.
Yeah.
Video, you want a partial zoom?
There's the lasers. Wow. Okay, that's what was pretty confusing. So it is two different sponges. The ones the lasers are on right now, kind of the leafy upper part, is probably in the family Phariidae, or was when it was alive. And this one on the lower side here could be a Pharynnematid, I think. So two different things. They just happened to fall together when they died. Or probably the leafy one fell on top of the other one. The other one probably didn't
If
fall.
it's possible to zoom in on those white branching structures on the surface of the mitt side, if you will. Do you see
on
them?
the right side?
Yeah, on the right side there are these
Okay,
tiny
let
little.
me turn a little bit. I see what you're looking
They
at.
look like those branching
Is that,
foraminiferans again to me.
Zoomed in all the way, video, or we partial?
Yeah, again.
Partial. You want to go ahead and zoom in? I'm going to turn this a little bit.
Here we go again with the funny little branching things. We don't know what they are. All right. So this might be something similar to what we saw before. These, I think we thought that they might have been bryozoans before, but some specialists had said that they were not. And so without an answer, I can't tell you what these funny little white branching things are.
All right, so I'll log this as who knows 4. So
These are so small, and the shape doesn't look to me like the bryozoan colonies, that I wonder if they're forams, but I can't tell. Are there little tentacular-like tips to them?
I guess you could call them that, yeah. Yeah, they're certainly foram, possibly
You want to
forams.
go back out wide? I'll see if I can get a little closer.
Do you want to get closer, or are you fine?
No, I think we're good.
We're
Don.
We're good
Thank
there?
you.
Okay.
Yeah, yeah.
Roger that.
Yeah.
Okay, then
Makes
we're heading on then.
Yeah. They're interesting, but we haven't seen another good-sized starfish today, so we need to find one.
I'm telling you, they're hiding. That's what it is.
They're not hiding.
Oh,
Could be
okay.
under that sponge.
They could well be. They do feed on them.
Attention all hands. There will be a spill response team meeting held in the Ford Lounge in 10 minutes. Again, that's a spill response team meeting held in the Ford Lounge in 10 minutes.
Got another Walterior sponge in the background there. Everything else is sort of dead around here, and we've already imaged the Walteria already.
What's that white thing that's on the branch?
Oh,
of
yeah,
the
we could
sponge?
take a look at that.
On the sponge
so-
to the right, the white.
Yeah, see that white sort of thing coming off the left side of the sponge?
I see it.
Yeah. And it's another black coral that could be Trisopathes in front of the sponge. This is
ROV nav bridge, move complete.
Thank you, Bridge.
Tina
it's
makes
a sponge, I think, a Munidopsis. I mean, a crab, I'm sorry.
Yeah.
Maybe a Munidopsis.
Tina makes an interesting point
So,
that
co-pilot,
she's
to be
joking,
on
but
pace,
you
should call in
the
next
the reason we
quarter
have so many
reports
bamboos is
from
because
now.
it's a beast-free seamount.
Way to
But,
go,
that
way to
would-
go,
there-
Tina.
It's-
She
Doesn't
may have an
seem
interesting point
unrealistic.
there that
Repeat your
there could,
last.
for whatever
Yes,
reason, there could literally be no sea stars
to
here,
keep on
which
pace,
might be
the
why so
next
many
30
of
meter move
these
should be in
colonial
about,
Didarians
2 or
are
3
so
minutes.
abundant.
Yeah, I think go ahead and get it.
I'm sure that'll be a
Yeah.
temporary
That's
situation,
an
however.
interesting
Sure.
topic to
So
follow,
like,
and I won't right
245?
now because we're looking at a squat lobster, but I am going to say that this looks to me like a type
230.
of muted squat lobster, not something that we
230,
usually
yeah.
see up on a coral or a sponge. So I'm a little bit surprised to see it perched in this location.
Yeah, Scott, this sure looks like Munidopsis to me. The very white, large size,
Bridge,
the very
right.
long spine
Another
coming
move,
out of
please.
it. And
30
if you look at the
meters,
front
bearing
claws, if they
230,
consume a little tighter on the
0.2
front
knots.
claws, they tend to be more robust in the
Range,
genus
30
Munidopsis
bearing
than
230
in some of the
degrees,
Chirostylids
steep,
and
0.4
Munida. And I
Copy.
think
Coming
that's,
up.
I would be willing to put at least 50 cents that it's a Munidopsis.
Yeah, Chris, and I also can see a little bit of orange behind that back leg. I'll bet this is a female, and she's got eggs, and perhaps she's hanging around up here to aerate the eggs.
There's a brittle star on there
That'd
too.
be kind of
Yeah.
interesting if they're in incubation mode. They actually try to climb up on things to do exactly what you're talking about, to provide more aeration, oxygenation to the eggs underneath their tail. And he's talking about right underneath the last leg there.
ROV nav bridge, move
Go ahead.
initiated.
I don't
You
think
said
so.
that. Sorry.
No, go ahead.
These ones?
You said this Munidopsis has more robust claws, which you can
Agreed.
see really nicely here. If I recall, Chirostylidae, the other kind of squat lobster that we were seeing associated with the Chrysogorgia corals and so on, they have very narrow chela, and I believe Chirostylid means something like needle or pincer hand, something like that. I'll have to check it out, but it's referring to that less robust and more pointy claw.
Yeah, and Tara Luke in the event log also agrees with the Munidopsis. I think Tina is also, so it looks like we have a consensus. So we're good for
Okay,
science.
we're good.
if you're-
Yes,
Okay,
thank you.
deal. When you're done.
To go back to this question about, where are the sea stars, and maybe that's why there's so many bamboos.
moving on.
It works both ways, of course. You wouldn't expect
Gotta
to see
move
the
it.
sea stars if they
230.
didn't have anything to feed on.
Copy that.
But where
I'm
there
already
would be an
there.
abundance of those predators, you would see the predation. So I don't know if this is a chicken and egg problem or what.
Oh, sure. I think it was just an interesting point that Tina had brought up, and it's something that isn't immediately obvious. The how do they say it? The presence of absence isn't the absence of presence.
Just come straight
Wait,
up.
Yeah, I'm agreeing with you, Chris. I do think it's interesting. That's why I wanted to talk about it a little bit further. And it's just interesting to speculate about why they are or are not present.
Yeah.
There's obviously other characteristics, other factors are involved just besides the presence of some food, some prey.
Oh, yeah. Their distribution. Sorry, Chris White.
No, I'm sorry. I was going to see if you could get that, whatever was in the water column, but it flew by already. Go ahead. Sorry.
Oh, no, that's fine. Yeah, I mean, there could easily be things like settlement. The larvae find, coral to settle on, or it could be that, there are just other conditions that are not conducive to development or that isolate them. There could be, we saw a fish earlier, maybe the sea stars themselves are fed upon by another predator that keeps them from sort of being established, if that's in fact, what the case is. But now that we've said that they're not here, we'll probably see a bunch of them. So give it 5 minutes.
Now you have sort of a coral-free zone, so at least they're giving you a break here, Chris, from corals, or you threaten them by going down and eating some yourself. So it's something's going on. A little more sedimentation here too, so maybe that's part of it.
Getting out there a little ways.
Yeah, I'm going
lateral. Yeah.
Sideways right
There you
go. Let's see. We had to move in.
Little
we haven't started to catch
reminder
up yet.
of what's going on here. We're on the
you
NOAA
just
ship Okeanos
are going a little quicker.
Okay.
We're on a Seamount
But
that
we're still,
is
Sirius still
tentatively
Yeah.
identified
Okay,
as
copy.
Sally T-Mount. That name is under
You're
review
also
right now.
drifting with the current.
And
There
our
is a little bit of current.
depth is
Okay.
2,100 and about 46 meters, 35 meters. And this cruise, we're on a cruise here. We're on a seamount just south of Johnson Atoll. The cruise is La Lima o Okamoana, which means partnership of the sea. And we've got a whole bunch of people in our event log helping us out.
You done?
I'm Chris Kelly. Next to me is Chris Ma. We got Nicola Rodriguez next to me. And then I think we got some new front row squatters here, if I'm not mistaken. I wonder if they could give their names when they get a chance.
Yeah, this is Dan Rogers, just temporarily filling in for Chris on co-pilot.
And watch Leadbird just moving a little sideways here, waiting for Sirius to catch up with us here.
ROV nav bridge, move complete.
Thank you, Bridge.
Alrighty. Oh, a dead sponge stump right, that black thing. There's another black coral that we tentatively identified as Trisopathes. These are some things that we've been seeing already today.
Pilots.
Chris Ma has a very distinct frown on his face next to me right here, so I'm thinking maybe we could crowdsource
What
the
was
search
the
for a
bearing?
sea star today. And if we can get everybody out there who's watching to keep their eyes wide open for a sea star and somehow get us a message that we've seen one, because I think
all right.
Chris needs all the help he can get.
We'll see one eventually.
We'd like to
I'm not
repeat
worried.
that last move, 30 meters,
It's got-
bearing 230, 0.2.
I may be too late. I was going to say, if you just
30
started
meters,
hanging out here waiting for
30,
Sirius, there was a black blob
coming up.
on the bottom, but it's now back some ways to the right.
Oh yeah, I saw that. I think that might have just been a rock
Okay, thank you.
or a broken face of the basalt.
There's another one of these Semperella sponges, that white one with the very frilly base. That'll be coming up shortly in your view. The black coral's right in front of us in the laser right now. Again, you'll see it in a few minutes, those of you who have a bit of a delay.
ROV nav bridge,
So Chris,
move
the
initiated.
Semperella I noticed is sitting on a massive brown mound.
Yeah.
What
Maybe we could
Does
get
it-
a zoom on that pilot if we're waiting here for a moment.
Sure,
Right
we
what
there.
is that? Does it make it?
Video
Is it
can
a
be
special holdfast or something?
Yeah. So this is a sponge in the family Pheronematidae, and one of the diagnostic characters for this family is the way they attach to the substrate. The family attaches by this whole network of fibers that are called well, they're called basalia. They're modified spicules, which are filaments. And oftentimes you see sponges in the sediment because they can anchor themselves, just like a plant anchors itself in the soil. These sponges can anchor themselves in the sediment. But every once in a while you see these sponges that have managed to grapple a hold of rocks, even though it seems like their roots are more appropriate for sediment type of living, and this is an example of that. So that's how it stays upright and holds on to its...
wow, that's a big cluster of glass, that's for sure.
Yeah, and it's very distinctive of this genus Semperella. Some of the other pharenematids have a much more reduced array of basalia, the attachment spicules.
It also looks like you have a shrimp hiding amongst the sort of spicules there.
Is that right? Oh, the red thing.
Yeah.
Yep.
So it looks like those
Zoom
fibers
in a
can
little
be
more video, please.
a habitat for other animals
Shrimp
as well.
in there. Stand by. Okay, is that good, watch lead?
He's distracted.
Yeah, got it.
I saw that, thank you.
Okay. Watch lead, are we happy with this?
Oh yes, please. Let's move on.
Moving on. Okay.
So Scott just had a question from the ROV lead here, Dan Rogers. So you happy with the progress here, or you want to keep meandering around and zooming in on interesting things, or would you like us to make a little bit faster tracks? Well, what's your desire here since you proposed this dive?
Yeah, I think we're seeing repeated patterns of the same taxa, so let's try to make some headway, get a little farther up slope, and see if it changes as we get to shallower part of the ridge.
Copy that.
Great. The pilot copies that, so thank you, Scott.
Okay, and we have the move in already,
Yes,
right now?
Yep.
Okay.
The last maybe 20 or 30 minutes we've been...
Chris, this may be an Acanella right here that now that I've said to move.
We just keep this up, we'll be good.
Trichotoma?
Yeah.
Put another one in at 230.
No, not trichotoma.
There's
Yeah,
another one at
trichotoma.
230. I put that in.
I
4 minutes ago. It should be
think
almost
they just
done.
went by it,
Perfect.
Scott. I'll keep my eye out for it. I saw what you're looking at. I just wasn't pondering it. I was actually looking at this rise of rocks over on the left. But yeah, I'll see if we can find another one.
What was it that he was looking for?
It's
Watching?
funny because as we got closer, it may actually have been a primnoid, but it had that whirl, that burst of branches from one point.
Okay. Anyway, I know that if we do see an Acanella, I know what they look like. So so pilot, is that? No, that's a primnoid. Sorry, excuse me. Just trying to see if I can find one similar to what he was talking about.
Looks like we got
This
a
is some
little
more
bit
of
a
these
ridge here.
Lefroyella glass sponges. It's a little bit different. They form these vases on stalks, which are really interesting.
And Chris, I said trichotoma. I meant Weberi,
Yeah.
Acanella Weberi.
Right.
I don't actually believe it's truly an Acanella. That is how it is classified, but the
Same
genetic
shoot, and
studies
looks like
say
it
that it
goes
is
up
nowhere
over
near
here.
Acanella.
Is that something in the water column there? Yeah, Sirius, or is that a...
No, that's the sponge on the
Sponge
bottom, but
on
you
the
do have
bottom,
some sort of
okay.
ridge
Yeah,
dead ahead
I see
of me
it.
here. Yeah, you're pulling me, so why don't you come back center screen here?
So,
that.
pilot, I hate to request a another pause here, but
Okay.
there look like there are a lot of collectible rocks here, and it'd
Okay.
be nice to get two. This is as good a spot as any to possibly grab another rock. We're looking
We
again.
may have to wait just a minute for Sirius to catch up here, because we're pulling
We are
him right at the
moving,
moment.
so
Oh,
should
okay.
catch up
Then
soon.
this might be a
Just
good.
stand by.
Yeah, this might be a good place then.
chip is almost done with that move.
Okay.
So
Sorry, Scott,
we
is
should
that
actually be pretty
that'll work
good.
for you just to get our second rock here, and that'll be our last
You've-
rock
you've got
collection
enough
for
to
the day?
Go down there and set up. though,
Set
if you
up,
want.
yeah. Okay.
Yeah, I have no problem with stopping for a rock. I guess one question I would ask you is, are we at a significantly
ROV
different
nav
depth
bridge.
compared to where you got this rock, or does that not matter? We want to have double the chance that we get a good rock as opposed to just some consolidated sediment.
Yeah, the latter, Scott. These really, really old seamounts, it's very difficult to get a rock that has basalt in it, so I think we're hedging our bets by getting two, and it doesn't matter whether one's at a slightly different depth than the other.
Yep, yeah, I know it. I agree. It's usually so consolidated that it's hard to find anything broken up. So, yeah, please take advantage of what we see here.
Just go ahead and set up on one.
There are,
Great, thank you.
I know we're looking
I wonder
at
if
rocks
you get a
now.
chance. Can
There
you
are
back up just a little bit and turn to the right? Was what looked like a pretty large Eurytigorgia magnus by rail as it just went by the edge of the image, and it would be nice to confirm that.
We're pretty far past that, actually.
Yeah, so I don't know. We can ask. Saw a coral as we were approaching this rock pile, but maybe we can after we grab the rock.
Yeah.
Okay. Yeah, he's gonna take a look, Scott, after we grab a rock.
Lasers on?
Yeah, they're in the. I can see them. A lot of these are
Oh, yeah, I
pretty
got
big.
him. Yeah, I see him again. Yeah. Let's see. What are we thinking here? Maybe this one in between the two big ones.
Yeah. What about that
Which one?
See, no, I lost it.
I'm looking at this one here. Stand by. Just kind of jammed in between these two, right? The laser just on it now.
I think that's going to be glued in
Glued
there,
in,
Don.
you think?
Yeah.
Okay.
Wonder we can.
Let's see what else.
So
we
there's
got.
some possible angular ones that are just a little bit further upslope.
Okay.
In fact, this is probably too big, but that's clear broken pillow, I think, over on the right. I think it's too big.
Oh, I see what you next to the stalk that's
Yeah.
laying down there.
That's it. It is, isn't
Yeah.
it? Too big.
Chris, what do you think? That looks a little big, doesn't it?
The one at the...
Yeah, stand by the lasers. You see that stalk going up there?
Yeah, and then this
You might be able to go,
dark
huh?
face there.
And
Let's see.
May not even be
Yeah.
free, but
Think that one's all right? Right there, the laser on it now.
Yeah, let's go for it.
Okay.
that's very likely to be basalt, I think.
and if not that one, there's an angular one
Yeah,
to the
that's
left,
good.
just going off screen. But let's try this one, maybe.
Let's try this. Let's see. All right, so that's a good position for us there.
I'm going to change the phases real quick just
All
to
right.
get a better view on depth.
Yeah, watch,
Yeah.
especially that tether with the
What do you
ways
think?
pointed.
Yeah.
We're okay.
Yeah, just
Yeah.
when did the ship move in?
3-4 minutes ago.
Okay.
I think you'll end up right above him.
Okay, and we're going to want this in the other box. You want to keep them separated, or does it not matter?
Let's try to separate them if we can.
All right. Okay. Let's
Complicates things because you got to open the lid. Sorry.
It's okay. It's good practice for me. I hadn't done that yet.
Okay, you're all spun up on hydraulics now.
Okay.
I've got to keep an eye on Syria, so I'll let you do
All right.
the rest here. We're kind of cutting it close with this ridge here.
You think?
So,
Okay. The mini Zeus up there on the big screen, though, so I can open
Yeah.
the lid. And maybe, yeah, that's great.
what was that now?
I didn't realize that ridge came up so much.
Yeah.
All right. Wrist is on. Our hydraulics on. And let's.
This one's going to be port outboard.
Yeah, it'll be port.
Yeah. Sorry, I don't mean port outboard.
Port forward.
Forward rock box.
Maybe give it a poke first.
That's it.
Oh, yeah, that's a good idea. Poke the rock before we go messing with the rest of it.
Come on, baby. Come on, you can move. You can wiggle. We're going to give you the most thrilling ride of your existence here. Now, come on, move. If you just move. It moved.
Okay,
He's going to get a ride.
All right, let's go.
Dave, why don't you back us up a
You know,
little?
that sounds like it's ridiculous overkill, but so many of these rocks are glued into the substrate. To actually see one move is really exciting for me.
I got you.
I have no doubt of that.
Does that help you?
You think
Not
10
sure.
meters is enough, co-pilot?
Come again?
10 meters enough for that backwards move?
I think so, yeah.
Bridge, nav.
This is the bridge. Go ahead.
We get another ship move, please. 10 meters, bearing 050, speed 0.2.
10 meters range, bearing 050.
Let me bring you in a little. Just hold there.
0.2.
Good copy.
Coming up.
Great, thank you.
So, Don, I think you are lying that you never did this before. You popped
don't
that thing
worry.
open
Yeah.
like an expert.
Don't,
Yeah.
Don't force that
Yeah, I
open.
thought that fell all the way open.
It does. It was hitting.
Hitting something over there. Okay, all right. Let's get back to where I need to be here now. Okay.
ROV nav bridge. Move initiated.
Thank you, Bridge.
I think I'm going to have to grab it at this end, right?
I think you're gonna want to go a little bit more in the middle. You look on mini zoos.
You think you can even, let's see.
Yeah, even if you get the fingers
Yeah.
past the. I'm gonna turn my head to give you a little more southern.
Oh, yeah, now that is a good rock. That,
Oh, and it conveniently is itself for our box.
oh, did it break?
Yeah.
No, I think that was a separate rock that was
Yeah.
leaning
Lying
against it.
against it.
Yeah, maybe.
It
Well,
didn't
either
break.
way.
Yeah. Yeah, let's see. Maybe there's a baby sea star on it. You never know.
There's a bunch of white stuff on it, to be sure.
Yeah. It's a very curiously shaped rock. So,
Okay, so
but with any luck, we've got something inside of it.
pretty good size. Okay,
Alrighty, thank you.
you're welcome.
ROV nav bridge. Move complete.
Thank you, Bridge. Should be feeling that backwards motion.
Yeah.
Oh, that's big, Chris. You don't mind carrying that up the stairs, do you, from the RV?
I've carried worse.
Oh, my back. It's just feeling a little bit sore right now.
Come down for you. Sorry.
You know, worst comes to worst, we take that gray bin and we grab an end of that thing. That's how we've gotten them up before, these big ones.
Just ask Sean.
That's fine. Will give us a chance to work off all of that food we've been eating.
Nice.
Okay.
Fantastic. Thank you, Don. It's going to give us a chance to find out how old Sally really is, although I know that females don't really like to talk about their age, but we want to find out their age anyway.
Oh, that's great. Oops.
You might need to nudge it a little.
Yeah. All right.
There.
What is it hanging up on?
It's a shilling.
You got it?
No.
Okay, thank you.
So,
Close these jaws.
All right, John Smith just made a comment in the event log. Good rock sample. Oh, that is great. Don, you pleased the geologist.
Very good. Thank you.
And there are a lot that's hard to please quite often. So,
you're feeling the rolls.
Did you know that Chris
You're
Maul
feeling the
got
rolls.
his degree in geology?
Yes, that's true. I'm a closet geologist. My PhD's in paleontology, so I don't know a whole lot about hard, quote-unquote, hard rock geology.
Alright, I think it's stuck on this line. Let's try it again here. See if you can just nudge it from there.
Move the shilling, maybe?
Yeah, I should be able to nudge it with the craft. If not, I'll do shilling.
Just
I'm not thrilled about where the
closed,
vehicles are, so if this is going to take a little bit,
you're not
we
watching
should
what's
move.
happening on the feed 3, I think. So.
There we go.
There it goes.
Okay.
Sort of.
Yeah, I think the rock is sticking up a bit.
Yeah.
Rocks.
Will that still work?
Yeah.
Okay, great.
Hey, Chris, this is Jonathan Tree over at UH.
Hey, Jonathan. How you doing? I'm glad you're calling in.
So, yeah, I thought I'd take the opportunity, since you just collected your second rock sample, to maybe add my 2 cents to the geological explanation that you've been long awaiting for the whole dive.
Thank you. Go for it, Jonathan.
So, yeah, so John called in earlier and gave a nice little summary of the unofficial name that was Sally. So as far as the geology goes, what these rocks are being collected for, and what a lot of them are going to go into for geochemical analyses, will be their geochemical compositions, which tell us a little bit about the mechanisms in which cause these volcanoes to erupt, and we can also back out an actual age from the geochemical analyses as well. So as far as the age of the Seamount goes, back in 2002, some new high-resolution argon radiometric ages were done. I think the rock that was actually collected that has the most relevant age for this seamount was very nearby, was dated around 71 million years old. So the weird thing about Johnston Atoll and the Line Islands themselves, so for those of you who don't know, Johnston Atoll lies at the northern portion of a 4,000 kilometer long submarine volcanic ridge. This ridge has 2 subparallel submarine ridges that compose the entire Line Islands. But unlike Hawaii, which has a very easily interpreted linear age progression, where the islands from the island of Hawaii, where Kilauea is currently erupting, is the youngest, going to the oldest in the most northwestern portion, and even older as they go north into the Emperor Seamounts. The Line Islands don't share that same age progression. So any more rocks that we can actually obtain and actually get geochemical analyses and ages for these will start piecing together a very complex history for this area. It's kind of weird. So the side of the Line Islands that Johnston Atoll is generally younger than the most eastern line chain. And so these two linear or subparallel submarine ridges share some of the same ages for distances of up to 1,200 kilometers, suggesting that there were volcanoes that were erupting coevally at the same exact time over distances of over 1,000 kilometers apart. So this kind of rules out the notion that a hotspot volcanic progression is the mechanism of choice for forming the Line Islands. And instead, we think that what is going on is decompressional melting of a heterogeneous mantle, which gives us different chemical compositions along the entire chain that are observed, and some of the weird age progressions or lack thereof that also are observed in the age data.
Yeah, thank you, Jonathan. I think the only, as far as I went, is that the geology was very complex in general in this area, and I think you definitely elaborated on that whole point. It's not a standard progression like the Hawaiian Islands. In fact, it's quite complex, as you were pointing out.
Okay, you can stop pushing out.
Rocks
so to give you guys a better idea what I say when I say decompressional melting of a heterogeneous mantle, that kind of thing takes place in the longest volcanic chains on Earth, which are the mid-ocean ridges. That's what generates all of the ocean floor in which these volcanoes erupted on top of. So when the actual lithosphere above the mantle is spreading apart, that leads to decompression. And when hot material decompresses, it goes above its melting point. And so that's what generates all the magma. So that's the first mechanism that we think is generating most of the magma in this area. The other mechanism, which could be contributing a secondary portion to the magma supply, would be these localized extensional features such as pre-existing weaknesses in the crust, like cracks that were actually formed at these mid-ocean ridges, and then transported along the lithospheric belt, so to speak, over to this localized decompressional melting. So the melt, just like a river, goes downhill, so it follows the path of least resistance is what you have to keep in mind. And these pre-existing cracks in the lithosphere are preferential localizations where magma can then transport itself and erupt at the actual surface. So it's a very complex interaction of lots of different geological processes, and yeah, like you said, much different than the original simple idea of Hawaii and the hotspot model, which generates these long island chains that are sort of comparative in the same distance, but not in the same mechanism. So it's very important that we make good collections and multiple collections in areas like this where there haven't been very many collections at all. So every single rock is going to be invaluable to the geological community once they've been once they've had their chemical analyses completed, and some of that data becomes available to the public for any other interpretations that are possible.
What is that small red thing to the left there?
Yeah, so Jonathan, thank you very much, and we will definitely continue to collect rocks as we go from seamount to seamount here.
Park it here for you.
And as you know, I always like to try to grab 2 rocks in case one of them didn't turn out so good. If you can call in every once in a while, if you see a rock that looks really good, or an area where we might be able to get a really good rock as we proceed on each of these dives, when you have a chance, that would be terrific. Otherwise, we'll do our best to get you a good one. Get all of you folks a good one, that is, all the geologists who are interested in this set of seamounts.
Looks kind of shrimp-like.
Yeah, so thanks very much for your comments. And we're doing a little bit of musical chairs here in front. Again, Dan thought that the back of his chair over in the co-pilot position was not as comfortable, so he made Dave move, and he bumped him and went over to the nav chair. So I think that's Dave up there is piloting, aren't you, Dave?
Yep, that's me, and that's Chris Ritter to my left now is nav.
Was it? It's Chris? It's not Dan. All right, it's Chris. Sorry.
And that's Don over in co-pilot. Same 3, just shuffled a little bit.
So Dan was like in and out real quick. Is that right earlier?
Yeah,
Ah, okay.
I've been mistaken for worse. So.
Okay, our current depth is 2,140 meters.
And we have a move in right now, right?
Oxygen levels are currently up over 2. They're about
Okay.
2.1. I just point this out
3
because
minutes
oxygen
in.
is kind of
Copy
a big
that.
deal here in the Johnston Atoll area. Some of you may not have heard the initial pre-dive
Yes,
comments,
pen
but
there.
we are going to have a mid-water dive at the end of this benthic portion. We're going to go up and do a set of mid-water transects from 800 to 300 meters, starting at 800, I think 700, 600 every 100 meters. Is that right, Chris?
I believe so.
Yeah. And
What was their time off bottom to do that today?
It's
we're going to see some
Fernando
really cool
calculated, it's all
stuff,
out
some
there.
weird jellies,
Video, can we
completely
zoom on
different
this?
community that we're seeing here on the bottom. And so those of you who are interested in all those jellies and snaggletooth fish and things
black
like
coral.
that, hang on, because we're going to have an exciting show at the end of this dive. We got a black coral right now, and has Scott fed his dogs, and he's back on, hopefully. Yeah, so he is back on. We do have a question, Pilot, as to what our off-bottom time is today.
Yeah, stand by one watch, so I'll get that for you.
And we're waiting to see. Scott thinks this is a heteropathes. Thank you very much,
This is,
Scott.
1537 or something. One of those many post-its has it. All right, video. That should do it. Thank you.
What was the last heading that you gave the ship?
230.
230. Copy that. Thank you.
I hear you right, Pilot. Is about 3:30 or so. We're going to be off bottom. Is that correct?
Nav's going to check that for me, but that was
Affirmative.
my recollection.
Yep,
Okay,
1530
thank you.
local.
And we're going off a little bit early so we can actually do these mid-water transects at the end of this dive. Got one of these stocked euplectellid sponges, this big yellow thing, which is in the subfamily Bolosomini. And possibly another bamboo on below that.
I normally straighten up when I get in there, but no, none of it's mine, but I normally straighten it up anyways. Not sure if I have quite enough tether to get out to this.
I can
Got
come down a little bit,
Gorgia
give you
coming
some.
up. One of these beautiful chrysogorgid corals that we've seen in many places.
I'll
These are
try it.
These are always
Yeah, you
beautiful.
should get there.
Sometimes they have interesting animals on them too. So the little bit, we're in a little bit of a flat patch, and then we're going to go up again. The contours are going to tighten up.
Move complete.
They're up, and it'll get a bit steeper.
That move just stopped. Let me know
Copy that.
if you guys want another one.
So
I
Scott,
think we're ready for it.
Scott was
Right.
wondering whether that
230
was a
still good?
Pinoet. I think it could be.
Let's see.
Was that
Yeah.
something that we can get to now that you were?
I don't think so. I think we can just move on.
Okay.
It's kind of hard to tell.
I think
Just
the
flat there. We've got
consensus is
targets
sort of to make a few
230
more
to
tracks unless we see something
probably
really unusual.
250.
We're
Yeah,
keeping
shoot
the ship
230. We'll
Great,
see.
Thank you.
Okay. Copy that.
Bridge, go ahead.
Can we do another ship move, please? Range 30 meters, bearing 230 degrees, speed 0.2 knots.
So
Range
Les
30
Watling
meters, bearing 230 degrees,
went ahead and
speed
concurred that we
0.2
had a
knots.
Rita Magnet that Curl Q1
I already
that we
saw
saw
that one.
a few minutes ago. So a little Bathopathes we just passed on the bottom, a little black coral. It's been a bunch of black corals today.
ROV nav, move initiated.
Copy, bridge.
So my co-lead Chris Ma has taken a quick break here, so all the sea stars out there, it's safe to come out if you guys want to come out now.
Is that one up there?
That white thing,
Yeah.
I think it's possibly a stock sponge. Oh, there you are. It is. They're coming out. He's left the room, and there's a sea star. I knew he was inhibiting them. Yeah, we would definitely like to take a look at this, please.
All right. If someone
I
can keep an eye
knew
on the door, we can lift off as soon as he.
They were
He's back.
hiding, right? I told you.
All right, video, come in.
Chris, we're zooming it right now.
I'm getting a little bit of a
So you
tether
guys
tug. I
won't
don't
believe
think there's
this,
much
but
you can do about that.
Out of the control room, after all day, we see a starfish. So this is Henricia, and I just.
There you go. I think you're now. Sorry.
All right. So for those of you
Let's,
who might have missed that,
zoom in more. We'll get some
I
details
literally
here.
walked out of the control room after all day and missed the starfish. So this is Henricia. This is a genus of sea stars that is actually found all over the world, sometimes in the Arctic, the Antarctic, and in the deeps. These are actually really hard to identify because they all look alike. And so there are people currently working on this problem. But they're ecologically very significant. They often feed on sponges. They're often animals that feed on sort of microdetritus and other things. But you'll often see them in great abundance in some areas. So it's a good thing that we found one on this very coral-intensive dive, and I'm sure the next time we see a sea star, it'll be feeding on something, which I expect is going to be the case given all of the other critters that we're seeing. So anyway, thank you. We can move on.
All right, come on out.
I was verbally abusive to you, by the way, when you're gone and we saw the star. I basically said you were inhibitory, and I told the stars to come out, that it was safe to come out while you were gone.
So it's your fault.
No, it's like they knew you were here looking for them. Anyway, that was funny. That was
Thanks. That's sort of like yesterday, the fish bombed me while I'm talking about the sea stars, and they go into hiding. It's just not fair. Oh well.
This is a big farnematid sponge that's sticking out the right side of that rock. And a little bit different member of the family Farnematidae than the Semperella that we were talking about earlier.
Do we want a close-up of that?
Maybe a snap zoom would be fine.
All right, we'll
These
try
are-
that. Video, come on in. I'll try to stop our motion
Yeah.
here.
So this is in the genus Polyopodon. And here you can see how it's attaching to the rock. It's using that very fibrous attachment. And that's how it holds onto the rock. It actually can grip the rock very tightly with those fibers, and that may even be a better way of holding onto the rocks than gluing onto the rocks like other sponges do.
It's worth pointing out, too, that those aren't fiber. They might be fiber-like in appearance, but they're actually more like glass. Those are sort of more akin structurally to fiber optic cables in terms of how they feel and how they appear. Weird-looking animals.
All right, video, that should do it. And I will get back to the center here. Is that a fish? It's up too high.
ROV nav bridge.
Yeah, the
move
That
fish
little
is way up there.
stock
Copy, thank you,
sponge
Brad.
is a little different. If
The one near the middle?
Yeah, right here in the middle, it's
All right.
bobbing. Just
Okay,
again,
guys.
snap zoom. It looks like it's one of the common ones that we've seen in many places.
Move complete.
Copy.
Is where,
Okay, video, snap zoom on this.
yeah, so this is Calophacus. It's in the family
Coming
Rosellidae,
back to it.
but it's in a very different family than some of the other stock
Hey,
sponges we've
Chris.
seen. Yeah.
What's that interesting? Is that there's a little crown-like structure in the background to the right there?
It's
Is
probably
that
a dead cup coral or
a
something,
coral?
maybe.
Ah,
Yeah.
okay.
And so,
Video, you want to push to that? Maybe cup coral, sorry. I forgot how zoomed in we were.
You gonna go down to it?
I bet it's a
Trying to-
4-amp.
and I'll bet you're right. So it's good we zoomed in on it. It is not a dead cup coral. What's a foramer? You mean a xeno?
Yeah, or possibly. So one of the neater things that live down here are not actually animals per se. They're single-celled animal or organisms that are more akin to amoebas.
All right, let's come back
These
out. I'm
create
bouncing too much.
sometimes
for that.
large, relative to them, large sedimentary structures that are
I'm
sometimes
going to turn my head
bald,
so we can
and
see
in that
you there,
case,
Dave.
it's
I'm gonna
more
center
like
up.
a,
You're going to center up? Okay.
like a little
Roger
cup.
that.
And they can form like reticulate, or rather sort of tentacles, and they can actually eat other animals. As single-celled organisms, they can eat animals, which is really remarkable. But it's not clear exactly what made it, so it's hard to say, but it's probably something like that.
So we have another Ridogorgia spiralis we're passing over, and some primnoids, bamboos. Pretty much what most of this we've seen earlier today.
It's very difficult for me to pass in a Ridogorgia without stopping to zoom in, but in the
Yeah.
interest of time,
Sure, thank you. Bolosoma sponge. Two different types there, actually. We've seen both of them already today. There's Bolosoma knee. It's in the subfamily Bolosoma, and Bolosoma in the genus.
all right, I'll push out a little bit. We could probably do another.
Part of this rock is manganese
What do you think
crust,
in there,
it looks
Copilot?
like.
Yeah,
It's,
sure.
breaking up, and it gives you an idea how
think
this
Let's see, more
crust
like
can form on the rocks down here if you've got enough time.
240?
And because, as Jonathan had pointed out, the
240.
Seamount is somewhere around 70 million years
I
old.
see up around
It's had a lot of
here,
time.
in my sonar at least.
That would make sense.
Maybe just serial setting would be fine.
I think that
Maybe that's a
Les
little too
Watling
much.
has pointed out
let's
that
see.
he's seen
225
three size
maybe?
groups
Yeah,
of
okay,
the
sure.
Ridogorgia maglospiralis,
Look up
and he's
slope
suggesting
here.
that there might have been at least 3 settlement events
I'll keep it at 30, but let me know
that
if
have taken place
we need to
at the
cut it
site
short.
for that
Okay.
species. Settlement being events where animals actually